Hello everyone. In this small video, I'm going to give you a basic idea on how to identify cells under the light microscope. Well, studying cells and tissues under microscope which are not visible with your naked eye is called histology. So this is an eukaryotic animal cell, and if you can remember, there are so many structures and arrangements that you study in a cell. However, not all these structures can be seen with the light microscope. So this is a light microscope, which is easily available in a laboratory. And this is the common appearance that you will come across with the histological slides. So without a basic understanding, this will be just an art with pink and purple dots for you. And let's try to move a step ahead through this small guide. And also, this is another advantage of the use of light microscope, which is that you can see a wide area of a tissue. And using different magnifications, you can focus on a specific area and can have a better view with more details. This is an electron microscope. See, it's huge. There are basically two types of electron microscopes: transmission electron microscope and the scanning electron microscope. So as the name suggests, TEM shows a cross-section of the cell, and we can see all the tiny details of a cell through it. But SEM is showing the surface characteristics. So this is what we are going to review today. So, ready, set, go. This shows an animal cell in higher magnification under the light microscope. What we can clearly see is the nucleus as a central dot and the surrounding cytoplasm. The cell membrane is not visible, but with the margin of the cell cytoplasm, we can outline the cell membrane. In addition, the nucleus, we can also see the nucleolus in some cells. Also, granules can be seen, and the mitochondria, of course, but not very commonly. In our body, there are many different cell types. They are of different shapes and sizes. That itself is the guide that helps us to differentiate these under the light microscope. So, the basic principle is that you have to look for the nuclei and the cytoplasms when you look at a histology section, and then we can work out depending on the characteristic features we see there. See, the first cell is squamous in shape. It has a central nucleus, and the cytoplasm is wider around it than the periphery, giving space for the nucleus. The next is a cuboidal shape, and this one is columnar in shape. These are tissues with squamous cells. See the flattened nuclei and minimum cytoplasm around the nucleus. The top shows the cuboidal cells standing in a line. See the square shape of the cytoplasm and the round nuclei. The bottom shows a simple columnar epithelium. See the rectangular shape of the cytoplasm and the oval-shaped nuclei. The cell arrangement is also different in certain areas. So this purple color part here is a stratified epithelium. There are several cell layers, and these circular dots represents the nuclei of each cell. The bottom part is the connective tissue. The cells here are wide apart. See these blue dots. And in between, we have the ground substance, with collagen and many others in pink color. This again is a stratified columnar epithelium, and this is a bone section. See, the osteocytes or the bone cells are arranged in circular manner. These tiny black dots, rather thin lines, are the nuclei. And next is a cartilage. See, the clumps of cells are in lacunae. These are flattened fibroblasts. Note, although that the outline of the cytoplasm is not very clear, we still can get an idea about the cell shape by looking at the shape of the nucleus. This is skeletal muscle. They have long cylindrical cells with many nuclei, and these nuclei are flattened and pushed towards the periphery of the cell. Also, you have to keep in mind that we are looking at the cut sections of 3D structures. So, depending on the cut direction, the shapes can vary. See, this is the same tissue cut in a different direction, this way. It shows the cross sections of the muscle fibers. This is a glandular tissue, specifically a mucous acina cells. See the pale stained cytoplasm and of pyramidal shape, or rather triangular in shape. Note that the acina means a ball-like arrangement. So what we are looking here is the cut halves of the balls. The cells are pyramidal in shape, pointing towards the center. So this is a cut ball. And see, the nuclei are pushed towards the bottom. And this is an adipose tissue or fat cells. The cell is full of fat, and the nuclei again are pushed towards the periphery. This is nervous tissue. See, the neurons or the nerve cells are star-shaped. They have processors. And they have big round nucleus, and the nucleolus is also clearly visible. And these supporting cells are comparatively small and scattered around. And this is a serous acina cell. They also have round nuclei with the nucleolus visible. Again, it's an acina. So you can also see the granules in the cytoplasm, like these dotted appearance. These are blood cells. Note the nuclei and the sizes of the cells. Red blood cells, they do not have nuclei in their mature stage, but the white blood cells or the leukocytes, they have many different shapes and arrangements of the nuclei. See this kidney-shaped or lobular appearance, single nuclei like this. So they show many differences. Some cells may have some special features like cilia or microvilli. You can see cilia, but microvilli are much shorter than cilia, and it's not clearly differentiate one by one under the light microscope. So they will appear as a brush border over the cell layer. Apart from the morphology of the cells, we can use different stains to differentiate the different types of cells and tissues. H and E, or hematoxylin and eosin, is the most common stain you will come across. Pink and purple slides. Hematoxylin is a basic dye which stain acidic structures like DNA and RNA. So the parts that contain acidic content will stain in purple color, like here. For example, the nuclei. Eosine is an acidic dye which stains basic parts in pink or red color. For example, the cytoplasm. Alcian blue. It stains mucin in blue color. So the mucin producing goblet cells here have stained in blue color. This is toluidine blue stain. Next is reticulin stain. It stains the reticulin fibers in a connective tissue in black color. Masson's trichrome is another special stain which is used on connective tissue, especially for the collagen. So the collagen is stained in blue or green, and the nuclei too. Cytoplasms in this section, muscles and erythrocytes can get stained in red color. And this one is silver stain, which is used to stain cell processes. So now I hope you may have a better idea on the same slide we started with. So this is an Echani stained connective tissue. You see the blue-purple color cells scattered around and the erythrocytes in red color. This again shows a similar picture, and you can see these erythrocytes or the red blood cells. And also, if you see carefully, you can see this surrounding layer of cells, thin squamous cells. So that is a capillary. So this is only an initial basic guide for you, rather a quick guide. So I hope this was helpful for you in some extent. And I'm looking forward to see you with more and more guiding videos in the future. And I will stop for now. Thank you for watching. I'm Randilini.